Hi, my name is Samuel Torres Jr. and this is my modeling project video on mitosis for anatomy and physiology with Dr. Joyce Banks on Wednesdays and Fridays at 11 a.m. I did a book about mitosis. Let's take a look at it. A lot of you may be asking, what is mitosis? Mitosis is the division of a single cell that produces two identical daughter cells. Steps are there in mitosis. Mitosis has numerous steps. The first step is interphase. Then it goes to early prophase, which is this one. Then it goes to late prophase, which is this one. Then it goes to metaphase, which is this one. After that, it goes through anaphase, which is this one. Then it goes to telophase, which is this one. And finally, it goes through cytokinesis, which is this. So the first thing that occurs in mitosis is the interphase. Technically, it is not a stage of mitosis. During interphase, a cell performs all of its normal functions, and when necessary, it prepares for the cell division to start. This is an illustration of early prophase. Early prophase contains two pairs of centrioles, spindle fiber, and nucleus. During early prophase, the chromosomes coil so firmly that they appear as one individual structure. This is an illustration of late prophase, which contains chromosomes with two sister chromatids. During late prophase, DNA replication has already occurred, which result in two copies of each chromosome. The copies are called chromatids, which connect to its duplicate called centromere. Also during late prophase, kinetochores are the protein-bound areas of the centrosome that connect to the spindle fiber to form chromosomal microtubules. Illustration of metaphase, which contains chromosomal microtubules from the previous stage and the metaphase plate. Metaphase is when the spindle fibers enter the nuclear region and the chromatids become attached to them. Once attached, the chromatids move to a narrow space at the middle of the cell called the metaphase plate. Metaphase is complete when all of the chromatids are aligned at the metaphase plate. An illustration of anaphase. Anaphase is when the centrosome of each chromatid pair splits and the two chromatids get pulled apart. The two daughter chromosomes get pulled to opposite ends of the cell along the chromosomal microtubule. This is an illustration of telophase containing cleavage furrow. Telophase is when a new cell prepares to return to the interphase state. The nuclear membrane begins to reform as the nuclei enlarge and the chromosomes uncoil. This technically marks the end of mitosis. This is an illustration of cytokinesis, which is the physical separation of the cytoplasm into two identical daughter cells. Cytokinesis begins at the end of telophase with the formation of furrow. The completion of cytokinesis marks the end of cell division.